Today I'm going to be going through a paper from 2021. As you can see, it has a ton of authors. I'm just going to say Ambati et al., but predominantly led by Isabelle Arnoff and Emmanuel Mignot. Many of us are familiar with them throughout the KLS universe. And so I'm going to go through and do my best to describe um, the things that this paper is saying, along with describing some things in terms, some jargon, and describe what they mean. So not only, so my goal here is to describe the findings of the paper, but also provide a little bit of a learning experience for any listeners that don't know what some terms mean, so that going through this paper can make a little bit more sense. All right. So we know what KLS is. All right. This is just an abstract. All right. I highlighted this because I wanted to point out occasionally associated with megaphagia and sexual inhibition. So again, this was in 2021. In 2024, there was a study of almost 500 patients that only showed maybe a third. It was 32, 33% of patients that exhibited sexual disinhibition. So I do want to say that in 2023, there was an update to the diagnostic criteria. And so the old triad of megaphagia, sexual disinhibition, and hypersomnia, the requirement of those three is no longer required in order to be diagnosed with KLS. All right. But moving along, similarly, I do want to point out the authors state remarkably there's a complete absence of symptoms between episodes. That is not always the case. There are many patients that do continue to exhibit some longstanding effects or effects in between episodes, and that has been reflected in imaging studies as well, which I did do a previous video on. So if you'd like to visit some imaging studies in terms of perfusion and metabolism within the brain, go ahead and watch that. All right. But anyways, back to what this is saying. All right. So we see a couple terms. What this study did was they performed a GWAS, all right, a genome-wide association study. And that's a type of experimental design that basically uses the human genome as a set of DNA instructions, right? That's what the genome is. And it's looking for genetic variances that are more common in people with a particular condition than those without that condition. All right, so you compare the DNA of people with the disease to the DNA of people without the disease, right? And what you're looking for, or what ends up popping up, is what we call single nucleotide polymorphisms, SNPs, SNPs, as we often pronounce it. And that's basically like a typo. A SNP in a position of DNA is where there's like a letter of the genetic code that differs between people. So some people might have an A in a certain spot, while some people might have a G. And that, again, is like a typo in a book. Most typos don't really change the meaning of anything, but sometimes there is a frequency in a certain SNP in a given population of those with a condition compared to those without. And that's what the function and purpose of a GWAS study does. All right. So the researchers collected DNA from over 600 KLS patients, as we can see, 673, and also used or compared them with over 15,000 matched control individuals. Some of those are going to be friends, some of them are going to be parents, siblings, but ethnically matched controls. All right, so people without the condition is what they're compared to. Um, and before analyzing that data, the researchers needed to account for the fact that some cases came from different ethnic backgrounds, whether it be European, Asian, Middle Eastern, and so forth. And so people from different backgrounds naturally have different frequencies of genetic variances, right? And if that isn't accounted for, it can create false signals. And so that's what the purpose of doing something called principal component analysis is. So the study might incorrectly flag a variant when it's really just reflecting an ancestral difference. And so to handle that, they used a technique called principal component analysis, which is just a mathematical statistical method that takes complex genomic data from thousands of people and distills it down into a few key dimensions, principal components, that captures the major patterns of that variation, right? So that's what that's taking into account. They also checked for genomic inflation, which is basically like a quality control measure in the GWAS method. And that factor, which is often measured in lambda, that's that little symbol right there, measures the overall statistical results that are inflated beyond what would be expected by chance. All right, so a lambda value of 1.0 means that there's no inflation, which is very good. There being only 1.06, that's almost nothing. All right, so that's very good. It's a very good statistical measure to assess the GWAS. All right. So they performed a GWAS. They found various SNPs. All right. These SNPs in the study are described as RS67 and so on and so forth. All right. That's just the individual variation of a single base of DNA. All right. That's what they're referring to, and that's what they're labeling them as. All right. I mentioned linkage disequilibrium because that's a technical term. And so what that refers to is a tendency of nearby genetic variances to be inherited together as a package because they're physically close on the same chromosome. And are rarely separated during the shuffling of DNA that usually occurs in reproduction. All right, so because of linkage disequilibrium, when GWAS identifies a significant SNP, that SNP might not itself be the cause of the condition. It might be tagging along with the true causal variant nearby, being linked, if that makes any sense. All right. So these are a ton of numbers, a ton of words. And so what they found, what the GWAS identified as a variant, reached a significance value. All right, and that SNP was the RS7194785. All right, that's located at the 3' region, which is the tail end of a gene, and that gene being TRANK1, which is found on chromosome 3. Specifically, this variant falls in a region that produces usually non-coding RNA, which I highlighted here. And what that means is non-coding RNAs are stretches of DNA that are transcribed, read and copied, into RNA molecules, but they don't produce or code for proteins, non-coding. All right. And so for a long time, scientists thought that those regions that don't produce, that are non-coding, were like junk DNA because they don't produce proteins. So what's the functional benefit? Why are they there? But it's now known that non-coding RNAs do play a critical role in regulating gene expression, almost acting like dimmer switches for upregulating and downregulating DNA expression. In fact, over 90% of RNA produced in the human genome are non-coding. So, little fun fact there. And so the fact that KLS the KLS-associated variant that they found in the GWAS is found in a non-coding RNA potentially suggests that it might influence how the TRANK1 protein is produced rather than changing the protein itself, if that makes any sense. All right. So that was basically the major takeaway of that section. All right. So hopefully that makes a little bit of sense. Hopefully it provides a little bit of background. All right. So again, the major result from that GWAS section is that there was a KLS identified association at the TRANK1 gene locus of a polymorphism. All right. And TRANK1 is already associated and has a strong genetic risk, or is a strong genetic risk factor for other neuropsychiatric conditions like bipolar disorder and schizophrenia, possibly suggesting a shared vulnerability between these conditions. All right. Association with symptoms of polymorphisms. And so after identifying that TRANK1 variant, the researchers examined whether or not this variant was associated with a specific symptom. So a polymorphism, again, is simply a genetic variant that exists in a population. And in this context, we're referring to that given RS, various numbers, SNP. All right. So the researchers tested whether the KLS patients who carried that variant differed in their clinical presentation from those without. So they looked at various symptoms and clinical features like the severity of the hypersomnia, cognitive impairments, and so on and so forth. And critically, whether or not the patient had experienced a difficult birth or had a birth with obstetric complications. And what they found, or the main takeaway, again, these are all numbers, and I'm trying to shuffle through this and provide what this stuff means for you. The main result was that KLS cases carrying that variant had a significantly increased report of obstetric complications around birth compared to KLS cases without that variant. And that's a interesting finding because it suggests that the genetic variant and birth difficulties were not merely independent risk factors, that they could have appeared to interact with one another. All right, so the TRANK1 variant is not just associated in general with KLS, it's specifically associated with KLS patients who have experienced birth complications. All right, and this points to what we call a gene environmental interaction, where the gene variant and the environmental insult, in this case a difficult birth, work together to increase the disease risk. And that's something that I did a paper on after this paper, which I'm going to cover in another video. So take a look at that when it comes. All right. The TRANK1 association is birth year dependent. All right. So what they did was, interestingly, compare the birth years. And so the strength of the TRANK1 association had an association with when the patient was born. The researchers divided those cases by birth year, and they found that patients born were more recently, meaning as we can see, 90s to 2000s, compared to older or more recent years. So in more recent years is what they're saying, had a weaker association with that genetic variant. And so to test that, they did a Fisher's test of allelic counts. That's super fancy term for being a statistical method being used to determine whether there is a significant difference in how often a particular genetic variant occurs in two groups. In this case, KLS patients and controls. And the word allele refers to, or the word allelic refers to alleles, which are different versions of a gene or a SNP that a person can carry. All right. So the test counts how many risk alleles appear in cases versus controls and calculates the probability that any observed difference could have occurred by chance alone. And so it's particularly useful, this Fisher's test, when sample sizes are small or when the variant is being tested is relatively rare, because it takes into consideration the probabilities rather than relying on just plain approximations. And so what they found was when they applied that statistical method test separately to patients born in earlier decades versus more recent decades, the association with that genetic variant and KLS was strong in older birth cohorts, but weakened or disappeared in more recent ones, which is interesting. And so this finding is consistent with a improvement, or one could suggest that this could be due to an improvement in obstetric medical care over the last 40 years. If the TRANK1 variant only increases in KLS risk when combined with birth difficulties, then as birth difficulties become more rare, then fewer people carrying that condition would have the increased risk of KLS. So hopefully that makes sense. This would make that genetic signal appear weaker with those that were born more recently. And so that does suggest that the TRANK1 variant alone is not sufficient to cause KLS. It requires a second, third hit on the genome or on the patient to increase the risk and lead to the development of the condition. Hopefully that makes sense. All right. So the GWAS analysis follow-up on KLS patients with and without reports. All right. And so to test, so again, these are a bunch of words, a bunch of data, but to explain again, to test whether or not their initial findings can be confirmed, they gathered an independent follow-up sample of about, let's see here, 171 KLS patients, KLS cases. And so what they tested was the genetic variant that we've been talking about. They tested that in the entire follow-up group, which oddly didn't show as much statistical significance. And that's not unusual in genetic research. Replication samples are often smaller and therefore have less statistical power or the ability to detect a real effect. Because remember that they did the follow-up cohort with 171 cases. Meanwhile, the initial GWAS was over 500 cases. All right, so that could account for that. However, when they split up the follow-up sample based on those who reported birth difficulties, the association was then again found like before. And this OR equals 1.54. What that means is basically an OR of 1 point whatever means that those patients were blank percentage more likely to carry the risk variant than the controls. All right. So in that case, with that number, an OR of 1.54 means that patients were 54% more likely to carry that risk variant than the controls. And in the remaining cases without reported birth difficulties, the association with that variant was not found. All right, so the main takeaway from that section is that the main result from that section is that the TRANK1 variant, that SNP that we're talking about, was replicated as a risk factor for KLS only in the subset of patients who reported birth difficulties and not in the overall follow-up sample. And so this does continue to suggest that there could be a gene environment interaction model, meaning that the TRANK1 variant appears to increase the risk of KLS specifically in individuals who also experienced difficult birth. And so without that environmental trigger, the genetic variant alone is usually not enough to cause disease. And that's widely accepted and experienced in a lot of other conditions, all the way from schizophrenia to cancer models. It takes multiple factors to contribute to the development and onset of a condition, not just a genetic variant, right? So fine mapping of KLS signal across ethnicity. And so as again, we all come from different backgrounds, right? So as explained earlier, because of linkage disequilibrium, go back and rewind to see what that means. The identified SNP, that little typo, might not itself be the cause of the condition. It might be located near the causal variant and inherited along with it. And so fine mapping tries to narrow down the list of candidate causal variants. All right. And so fine mapping with respect to the ethnicity is what they're taking into account, right? And so the fine mapping across so for example, as we can see here, in populations of African ancestry, for example, linkage disequilibrium blocks, tend to be shorter, meaning variants are less tightly linked, which can help narrow down the region of interest. And so in populations with European or Asian ancestry, linkage disequilibrium blocks tend to be longer, meaning that more variants travel together and it's harder to distinguish which one of them is actually the causal variant. And so by comparing the association signal across the various ancestral groups in their sample, the researchers can somewhat refine which variants in the TRANK1 region are most likely to be functionally important. And so what they found, or their main result here, was that the fine mapping confirmed that the signal was centered around the 3' region, the tail end of the TRANK1 locus, and was consistent across multiple ethnic groups. All right. Polygenic risk scores. Okay. So there's a lot of words, a lot of data, and so I'm going to try to continue to parse through what some of this stuff means. All right, and so most complex diseases, as I kind of commented on a Facebook post before, are not caused by a single gene. They are caused, or but by a combined effect of many genetic variances plus the environmental stuff that we've talked about before, each contributing a small amount of risk. And so the polygenic risk score, here I have highlighted, captures this by adding up the effects of thousands of variants across the genome into a single number that estimates an individual's overall genetic predisposition to a condition. All right. So to calculate the polygenic risk score, researchers they took the results from the initial GWAS, which identified which variants were associated and how strongly, and used those basically as weights. For each person in a new independent sample, they count how many risk variances that person carries, multiplied by how much weight it carries, the effect size. And the sum of all these weighted variances is the person's polygenic risk score. And so a higher score means a higher genetic predisposition. All right, so hopefully I explained that well. The concept is, and I wanted to, again, as somebody's reading this, I just wanted to point out some jargon and continue to explain that. Genetic liability. And so the concept of genetic liability is closely related. Liability is just this construct used in genetics to describe the total underlying risk for a condition, like a spectrum of high to low risk. Everybody falls somewhere on the spectrum based upon their combination of genetic variances, environmental exposures, and random chance. People who cross a certain threshold on this spectrum develop the condition or tend to. And so the genetic liability refers to specifically the portion of this risk that comes from the genetics, if that makes sense. And so when researchers say genetic liability was increased, what they mean is that KLS patients on average carry more risk increasing genetic variances than the controls. And so, so hopefully that makes sense. The total observed scale heritability. So this was something that they attempted to estimate. And so again, here's a ton of words. Let me scroll up. And so heritability is basically the proportion of variation in a disease risk that can be attributed to genetic differences between people. So the observed scale refers to measuring heritability directly from the binary outcome. Has KLS, doesn't have KLS, as opposed to the liability scale that we previously mentioned, which transforms that measurement to account for the fact that the condition is rare. So for rare diseases like KLS, the observed scale heritability can look very different from the liability scale heritability because so few people are affected. And so the liability scale transformation that they've discussed here tries to adjust for that, providing a more interpretive estimate on how genetics do contribute to the risk in population. And so, with all that said, there's a lot of words. The researchers calculated the polygenic risk score in their follow-up sample of the 171 KLS cases and found that the genetic liability for KLS was increased in those cases compared to controls. And so the statistical measure, if you just look here, this pseudo R squared being, and what they found, or what they saw as you're reading, you'll see that this statistical measure was a pseudo R squared equals 0.15, I believe. Meaning that the polygenic risk score explained about 15% of the variation in KLS risk, which is arguably a substantial amount for a rare condition. And the p-value, some of us in school remember p-values being like 0.05 and stuff like that. And so the p-value of being less than 2 times 10 to the negative, 16th, 20th, whatever it says, indicates an extremely strong and reliable finding on that itself. And so the PRS, and so the threshold being 0.05, as mentioned here, again, it says threshold was 0.5, meaning that they included not just the strongly associated variants, but also many variants with weaker associations, which is standard practice because even individually weak variants can collectively contribute to a meaningful information. All right, and so the main takeaway from this risk score paragraph. All right. A lot of words. Hopefully, as you're reading that section, I helped clarify what a lot of those words meant. Even though the single gene variant did not replicate as much in the overall follow-up sample, the overall genetic architecture, meaning the combined effects of those variants across the genome, was distinguished in KLS cases compared to controls, which confirms that KLS has a genuine polygenic basis, meaning many genes can contribute. All right. Where are we at? Gene analysis gene pathway analysis. All right. And so I'm going to try to explain pathway enrichment, what that means, what magma means, what ROR alpha is, and so on and so forth. All right. So beyond looking at individual genes, the researchers, pardon me, my cat. Stop. The Beyond looking at individual genes, the researchers examined whether the variants associated with KLS tended to cluster in a particular biological pathway. All right, so whether or not groups of genes that work together carry out in a specific function of the body. All right. So that's what they mean by pathway enrichment analysis. All right, and so to do that, they used a tool called MAGMA, and that stands for multi-marker analysis of genome annotation. All right, and so it's a widely used computational tool that takes GWAS results and performs two key analyses. First, it integrates or combines the association signals from all of the SNPs or variances within each gene to produce a single gene level association score. Essentially asking, is this gene as a whole associated with the condition? Secondly, MAGMA, it performs gene set analyses, also called pathway enrichment analysis, which is what this was referring to here. And what that asks is, do the genes associated with KLS belong to the same biological pathway more often than would be explained by chance? And so it uses a statistical framework called regression-based, which makes it pretty good and fast compared to older methods that relied on time-consuming testing. And so how that works is imagine you have a list of 100 genes that showed some association with KLS. You then check whether or not these genes are disproportionately members of a known biological pathway. All right. And if, say, 15 of your 100 genes belong to, say, the circadian rhythm pathway, or a particular enzyme pathway, or lipids and their formation pathway, so on and so forth. If 15 of your 100 genes belong to a circadian rhythm pathway, or whatever it is, you may only expect 3 by chance, and that pathway is enriched, meaning that disease appears to be specific, or that disease appears to specifically involve that biological pathway. All right. So hopefully the meaning of that makes sense. And so the analysis revealed, or before I get into the result, upstream and downstream, what that, or in 1 KB, that's literally just saying the amount of bases upstream, meaning ahead, or downstream behind that particular SNP. All right. So all of that to say, is that the analysis revealed a significant enrichment of genes involved in circadian regulation, which is pretty important. All right, being a sleep-wake predominant condition. All right, and so which is pretty important, in which the to understand why that matters, there are many circadian clock genes that contribute to the function of the circadian rhythm. And so it operates with a bunch of feedback loops of genes and proteins, and at its core are two proteins. This isn't in this paper, but I'm going to say it anyways. Called clock, ironic, right? And BMAL1. All right, joined together to form a pair or a dimer that activates the expression of other circadian genes, including one called PER and CRY. And those proteins that are encoded then accumulate and feed back to help decrease that activity again, completing a cycle that approximately takes the 24 hours that we talk about in a circadian rhythm. And so that's a predominant loop that drives the genetic component of the circadian rhythm, which is in which I'm now currently today writing a study or a paper about that with respect to KLS. So hopefully I can get that continuing to move. All right, so keep a lookout. A second stabilizing loop, which is what these authors point out, involves two proteins called ROR alpha, which stands for retinoic acid receptor, related organ receptor alpha. All right. And Rev ERB alpha. All right. So these are transcription regulators. Transcription is taking DNA, turning it into RNA. And so that RNA can then produce things for the body. All right. So they regulate the transcription. All right. So proteins that control whether or not specific gene is turned on or off. And so just a little bit of background, because it doesn't really provide it here. ROR alpha activates the transcription of that gene that I spoke on before, BMLA1, which is important in circadian rhythm. While this Rev gene represses or silences it. And so together they create this push and pull mechanism that helps ensure the periodic expression of, here again, periodic expression of the BMAL1 gene. And so what that means is the BMLA1 levels rise and fall in the 24-hour rhythmic pattern. And without the stabilization of the other proteins, then the circadian clock would be haywire, unstable, and not precise. And so the fact that KLS-associated genes are enriched in that pathway makes biological sense for a condition characterized by disruptions of sleep and wakefulness. All right. And so main result is that magma pathway enrichment analysis identified significant enrichment of circadian regulation pathway genes amongst KLS-associated variants. All right. Hopefully that makes sense. I know this is going to be a long video. I'm sorry, but I'm trying to be thorough so that everything somewhat falls together in somebody's head. All right. Exome sequencing analysis. All right. Remember when I talked about non-coding RNA? Those produce introns, things that don't encode for proteins. Well, exomes do. Exomes do produce and encode for proteins. All right. So while the GWAS looked at the common genetic variants, those found in at least a few percent of the population in KLS. The researchers also performed genome or exome sequencing on a subset of KLS cases to look for rare variants. And so again, an exome is just a portion of the genome that contains instructions for making proteins. And although it only represents like 1 to 2% of the total genome, exomes contain the majority of known disease-causing mutations. And so the remaining, like over 90% of the genetic genome is non-coding for proteins, but they are regulatory also, like again, the dimmer switch. And so exome sequencing is a technique in the lab that reads the DNA code for the protein making genes in order to look for if there's any mutations or changes that might disrupt the protein that gets encoded its function. All right, and so unlike common variants that are found by GWAS, which individually have kind of small effects, the rare variants found by exome sequencing can sometimes have larger effects because the exomes are producing proteins that can be functionally relevant. And so the researchers sequenced the genomes of the exomes of KLS cases to search for a rare or potentially high impact mutation of an exome. They did not identify any given rare mutation that was clearly responsible for KLS in the exome for KLS across patients. So that's good in terms of clarity. And so that's consistent with with KLS being a complex polygenic condition influenced by many variants of small effects and other impacts rather than a single gene disease. All right. So there wasn't a smoking gun mutation that was found. All right. The LMOD3 genetic variant. All right. And so there was a previous study, I think in 2019, that reported a variant in a gene called LMOD3 that they associated with KLS and a large family with affected members, several affected members. That gene is a structural or encodes for a protein that is involved in the scaffolding of cells. And so they help regulate the structure of what we know as actin filaments, tiny cables that give cells their shape and structure and stability. All right, and it's expressed in the brain and has been contributed or has found to be found to be present in major brain structures relevant to the hypothalamus, thalamus, and brainstem. All right. However, this study is now saying, hey, we double checked that in our GWAS, and we didn't find that there was or they examined the LMOD3 region and did not find a significant association between KLS and the LMOD3 variants. All right. And so that doesn't necessarily mean that LMOD3 is irrelevant to KLS. It may play a role in specific families with high rare, high impact mutations. But it does suggest that the LMOD3 variants are not a common denominator of genetic risk. All right. So that's what that means. Gene mutation burden test in KLS. All right. And so this is a statistical method used to determine whether a particular gene carries more rare, potentially damaging mutations in KLS patients compared to controls. And so the logic behind that burden test like um, is like this. It's like any rare single mutation might be too uncommon to detect statistically on its own. But if a gene is truly involved in a disease, then many different rare mutations in that gene might individually increase risk. And so by collapsing all the rare variants within a gene into a single test, you're essentially asking, does this gene have more rare mutations overall in cases rather than controls? And so you increase your power to detect a gene's involvement. Kind of like think of it like checking whether a particular street has more potholes than the average street. So any single pothole might not statistically screw over your car, but if the street has significantly more potholes than other streets, then driving down it, there's more likely to be something wrong with your car because there's something wrong with the street. Um, and so there's a lot of genes mentioned here. And so we see the GABRG, we see the CORD-DC, so on and so forth. And so I'm going to kind of describe what some of them mean. All right. So here, let me see, the NRX-N2 gene. All right. So that encodes for neurexins, which are proteins found on the surface of nerve cells at the point in which they connect with others, the synapse. And they act like docking stations on on the sending side of that signal, helping to organize the connection between nerve cells. All right. There are neurexin genes and variants in neurexin genes that have been linked to things like autism, schizophrenia, and so forth. The GABRG2 gene, so that's a gamma. We've all heard of GABA before. All right, so this is a gene encoding or gene relevant, pardon me, it encodes a component of the GABA A receptor, which is like the brake pedal of the brain, right? So GABA is the primary inhibitory neurotransmitter in the brain, calming neural activity. And the GABA A receptor is the channel on the nerve cells that, when you activate GABA, allows for specifically chloride ions to flow in, making the cells less likely to fire, which is what the inhibitory effect is. All right. And so the subunit encoded by this GABRG2 gene is a critical component of that receptor. And mutations in that gene have been associated with things like epilepsy, which should make sense if you ruin the ability to inhibit firing. The CORD-DC1 gene, all right, that's relevant to cysteine and histidine, which are amino acids. All right. And so the gene encodes a protein that interacts with something called HSP90, which is a chaperone protein. And so the chaperone proteins help other proteins fold into the 3D shapes that they're supposed to take on, like a helper helping you put together an origami, making sure it's folded correctly. And so if the proteins misfold, they can malfunction. And so that gene that's mentioned helps regulate the activity of the chaperone. And it has been implicated in cell signaling pathways. Yeah, I think that's all I'm going to say about that. The AATK All right, that is an apoptosis associated tyrosine kinase. That means that's a lot of words, but again, that this gene encodes for a protein called kinase, which is an enzyme that is able to change the activity of something. All right, that is predominantly expressed in the brain, especially in the cerebellum and hippocampus. AATK is associated or is involved in two key processes: apoptosis, which is the natural telling the cell to die, programmed cell death, as we call it. All right, so it's the body's way of eliminating damaged or unnecessary cells. It's normal. And neurite extension, so the growth of the projections of nerve cells that allow them to connect with other cells. And so the genes that AATK's expression increases when neurons need to undergo apoptosis. And the kinase activity that gets promoted promotes that cell's death. And again, that is a normal process that we see occur. All right. CASP7, KAC, KSC, or not K, oh, CASP7. I'm starting to lose my voice here. That's a capsase. All right. So capsases are a family of enzymes that act almost like executioners of apoptosis. So AATK is associated with pro-apoptosis. CASP7 is associated with stopping that apoptosis. And so when a cell receives a signal to die, capsases are activated in a cascade, like a chain of dominoes, that dismantles the cell. And so caspase 7, which is what that gene is referring to, is classified as like an effector or executioner capsase, meaning it carries out like the final cell's demolition. I guess one could say. And so along with capsase 3 and 7, it's one of the two main executioner capsases that are together required for the efficient apoptotic cell death pathway, right? I believe now that I say that out loud, I think I just misspoke like 45 seconds ago. So pardon me. And so what they found with this gene burden analysis is that the burden test did not reveal that there was a single gene carrying a significantly less number of rare damaging mutations in KLS. And so again, that furthers the support for the conclusion that KLS is predominantly a polygenic condition, not a single gene. So most of this paper is talking about TRANK1, a single polymorphism nucleotide, or a single nucleotide polymorphism. But again, the researchers did a very good job of doing the methods and data analysis needed to conclude that KLS is not driven by a single gene mutation. All right, it's probably contributed by a cumulative effect of many common variances of small effect, rather than being caused by a rare high impact mutation in a single gene. All right, and the researchers point out that genes like the Norexin, like the GABA one, like the CORD one, like the capsase one, the ones that I was just talking about, could be possible or plausible candidates given their roles in synapse function, neuron inhibition, protein folding. All right, but none showed themselves alone a significant excess of rare mutations in KLS patients. All right, and so that was a lot. All right, so discussion, they tied back to a ton of data that they talked about. All right. Last pages of this document are explaining some of the methods and stuff that I talked about, hopefully and explained. So central finding is that the TRANK1 gene is associated with KLS, especially in patients with birth difficulties, specifically those with birth difficulties. And it kind of establishes KLS as part of the family of disorders, brain disorders, in which there's a genetic predisposition that interacts with environmental insults during birth to produce the condition. And so this is what we call a gene environment interaction model that's well established for other conditions. All right. The TRANK1 connection to bipolar disorder is particularly noteworthy because it may explain the clinical observation of like lithium being a classic treatment for bipolar disorder, but it's been shown to produce a positive effect in a lot of KLS patients that are on it. And the protein interactions or the protein interaction partners are something not discussed here, but are something that I talk about in my own paper on genetics from 2023 that I'm going to do a separate video on because it bounces right off of this paper that I'm discussing today. All right. They do mention some limitations. All right. Where was that discussion? I think they probably talk about it in the I didn't highlight it, but pardon me. But I remember the authors acknowledge that there's some points of limitation. One, that the TRANK1 association did not fully replicate in the complete overall follow-up sample. It was specifically significant in the subset with birth difficulties. All right, so that again kind of supports the idea that there's this gene environment interaction going on. All right. Second, the reports of birth difficulties were collected retrospectively, meaning from memory, often decades after the event, right? These are family and patients that are much later reporting that they had experienced a birth complication. And so that potentially introduces the possibility of bias. But I think that this paper did a great job statistically of doing their due diligence to try to parse through some of that. All right. Additionally, KLS is super rare. All right. There's like, what is it? 1 to, or 2 to 8 cases per million, right? And so there's anywhere between like 8,000 and 40,000 people in the world with it, which makes it inherently difficult to assemble a large enough data sample for genetic studies. However, this is to date the largest genetic study that we have on KLS, and it is a big study. And because that 673 cases in this study that we talked about is a huge collection effort over the 14 years that this study was talking about. All right, so I do want to point that out, that this was a collection effort of over 14 years of labs across the world. And so this wasn't research that was just done in a lab over the course of a year and published in 2 years. This took over a decade to put together. All right. And so future directions, maybe. And so this finding opened up several avenues for future research, right? Understanding how TRANK1 variants affect brain development and function could reveal therapeutic effects, right? The circadian pathway enrichment analysis that they found suggests that drugs targeting the circadian clock might be beneficial, which is probably why Dr. Mignot did a paper on a couple patients using Rameltian, which in theory should help a patient with their circadian rhythm, which I'm all about. All right, there's a lot of reason to do it. That's not me saying go take Romeltian. What I'm saying is honing in on your proper circadian rhythm is absolutely necessary. All right. Finally, future directions is that genetic databases continue to grow and become more diverse, which allows for future GWAS studies with larger sample sizes that might identify additional risk genes beyond TRANK1. So hopefully in the next 10 years, we're going to find out even more. And again, so hopefully I did a decent job, pardon me, of explaining a lot of the jargon that's involved in this paper, hopefully what they mean. And so as you read through this paper, you have a much more better understanding of what those terms mean, how they applied, and hopefully I successfully communicated the result and takeaways of this study. And so again, what I'm going to do next, my next video, is that I'm going to bounce right off of this on a paper of my own on the genetics to talk about the protein partners that could potentially be at play with TRANK1, specifically regarding the possible effect of that environmental interaction and insult leading to the development of the condition. All right. So be on the lookout. Hopefully this was of good help, and I'm going to hopefully try to continue doing more of these. All right. Feel free to comment if you have any questions or reach out to me in any other way. Have a good one.